All right, everybody, we're going to go ahead and get started. Thanks for being here, and for everybody online, I think you are more than are in the room, so thanks for being here as well. My name is Cinnamon Moffett. I'm the Research Program Manager here at Oregon State University's Hatfield Marine Science Center, located in Newport, Oregon, and this is Hatfield's Research Seminar, and so thank you all for being here. A couple of quick announcements. Most of the folks in the room, at least, are familiar faces, so you've heard this before. But everybody online, your mics, cameras, and screen shares are turned off, but we do have a volunteer named Rose here monitoring the chat. So you can put in any of your questions, either technical or for our speaker, into the chat, and we'll read those out either when it's relevant or at the end of today's talk. For folks in the room, remember, if you need to ask a question at the end, please raise your hand. I'll bring you the mic, or you can go up to the mic stand up there. We might move that down a little bit, but and that way everybody online can hear as well. We are recording, which I didn't do. Roseanne, can you start the record? Maybe. If not, I'll do that in a second. But we are recording today's event. We will in a second, and that can be, we're good to go. That will be posted on the past seminar page in a few days. So if you'd like to share this with colleagues that couldn't be here, please do that. Wanted to make a couple of quick announcements. We have a pretty exciting talk next week at seminar. It'll be on January 26th. We have Kaus. I'm going to say his name wrong, and I'm going to apologize to him even though he's not here. Reg Q. Meyer, who is with Integral Consulting, and is going to talk about potential effects of large-scale floating wind farms in the coastal upwelling current of the California current system. So that should be really interesting, and I'm excited to have him here. I also wanted to just remind folks that it's next week is the Big Blue Film Fest on the 27th and 28th. So if you haven't already, you're welcome to go to the website and buy your tickets and join us for some really cool films. And if you need information on any of our events, you go to HMSC's webpage, scroll to the bottom, and there's a calendar of events there with everything you need to log in. But why we're all here today is for Zoe, our speaker, and Zoe was invited by Jessica Miller, who's going to do our introduction. So I'm going to hand this off to Jessica.
Don't fall on the stage. Good afternoon, everyone, and people that aren't here, hopefully. I'm Jessica Miller, and I'm here at Hatfield, and it's my pleasure to introduce Zoe Almeida today. Zoe's been a postdoc here since March of 2021. She came to us in the middle of COVID and has jumped right in a project with Ben Laurel and myself looking at marine heat waves and their effects on Gulf of Alaska Pacific Cod. And Zoe comes to us from, she got a BA in biology in Scripps College in Claremont, California, and then went on to get a master's at Purdue in ecological science and engineering interdisciplinary graduate program. And then she went to the other OSU, Ohio State University, where she got a PhD focusing in on the early life history of fishes and their ecology, evolution, and organismal biology department. And pretty quickly arrived here. And it's been a pleasure to work with her. And we're sad to see her go because she has a permanent position at Cornell, starting like tomorrow practically, as a senior research associate at the Biological Field Station in Syracuse, New York. So she'll be going back. She's sticking with the effects of warm waters on fishes, but she'll be going back inland to freshwater fishes. But today she's going to tell you about some really nice work she's done, really diving into the effects of marine heat waves on Gulf of Alaska Pacific Cod.
All right, can everyone hear me pretty good? Okay. All right, thanks everyone for being here either in person or online and for your interest in my research. So I'm going to be talking today about the effects of some recent marine heat waves in the Gulf of Alaska. So these marine heat waves started in 2014-ish and persisted through 2016, and then things cooled off again in 2017 and 2018, and then warmed up again, and another heat wave event happened in 2019. Now, this might sound kind of familiar to people around here because these marine heat waves occurred in the North Pacific in general, and that first heat wave, that one between 2014 and 2016, that one has been colloquially termed the blob, so it's even in popular culture. But these marine heat waves had a pretty dramatic effect on the ecosystem, including affecting a previously very sustainable fishery population of Pacific cod. And so that it affected the population to an extent so that the fishery actually closed in 2020. So given the duration and extent of these marine heat waves, all Pacific cod life stages were affected. To give you an idea of what their life stages or what their life history is like, adults will spawn in demersal waters offshore in the winter, and then their eggs will hatch into larvae in the spring. And for the first couple days, first few days, larvae will continue to stay demersal, but then they'll raise in the water column and start eating pelagically, where they'll do that. There'll be pelagic larvae for a couple months before they then settle into nearshore nurseries as juveniles. And as I mentioned, given the duration and the extent of the marine heat wave, all of these life stages were affected by the marine heat waves. And we can that's been shown in a variety of studies previously. So previous studies have shown that the marine heat waves reduced the spawning habitat for Pacific cod, also reduced the overlap of the larvae with their pelagic prey. And then it's also reduced the juvenile recruitment to the nurseries. And interestingly, although there were fewer fish that were found in these nurseries, the ones that were found tended to be larger and in higher body condition after these marine heat waves than before. So due to some of these observations that we've seen, our project focuses in on these early life effects. And this first part of the project is really focusing in on those temperature relationships with phenology, size, and growth. So some of the potential causes for those observations where juveniles were a lot larger and in better condition in those nurseries, I guess there's a couple different ones that I'll cover here. And I'll talk about them in relation to this term we use in fisheries called which is size at age. So when we catch fish in a population, the size of that fish can tell us a lot about that individual and about that population, but we can get even more context for that if we know the age of the fish as well. And so one reason that juveniles might have been larger in those nurseries is that they're actually older. And so if they have earlier hatching dates, they would be older when we catch them in the nurseries. And previous studies have shown, and this is a pretty common phenomenon, that individuals that are exposed to warmer conditions actually will hatch out earlier, either due to changes in parental behavior or things like incubation duration, which is affected by temperature. Additionally, or another potential cause would be that individuals actually grow faster. So this is also expected with metabolic theory and things like the temperature size rule, where juvenile stages, when they're exposed to warmer conditions, they'll grow they'll grow faster, which could again be why individuals are larger at age. And so if individuals before the marine heat waves were exposed to cooler temperatures, they might be at about that point in their growth curve. And then if they're then exposed to warmer conditions since the marine heat waves had occurred, they could be at that higher point and thus have a higher growth rate. And then finally, it could be that individuals are both older and faster growing. And so it could be that those warmer conditions since marine heat waves caused individuals to hatch out earlier and then grow faster. But understanding the effects of these temperatures requires us to disentangle some of these relationships between temperature, phenology, growth, and size. So heat waves cause warmer temperatures, and then those temperatures can affect growth rates through metabolic rate, and growth rates affect the size of individuals, which is also affected by how long those individuals have been growing. So we get that size at age kind of relationship. But temperature also affects hatch dates through incubation time. And then hatch dates obviously affect ages, and then hatch stage can affect growth through other environmental factors. So if individuals are hatched out earlier in the year, they're going to experience a different environment than if they hatch out later in the year. And then finally, we have to consider the effect of age on growth. So as individuals are growing and developing, they're going to grow at different rates depending on what age and what stage in development they're in. And so to really address these questions, we have to kind of tease apart the effects of these different factors. So our objectives with this part of the study was to quantify the relative importance of temperature on hatch phenology, size at age, and early growth. So today I'll walk through the collections that we have and the data that we use to address these questions. And then I'll walk through each of these analyses. So I'll walk through the hatch phenology, our size at age analysis, and our early growth, and discuss those results for each of those, and then the consequences, what it might mean that we're seeing these different responses. And then I'll wrap up by sort of concluding with what was that effective temperature that we observed. All right, so to start out with, I'll talk about the data that we used. So we used juvenile Pacific Cod that were collected in July of their first year of life in areas near Kodiak Island. And they were collected with beach seining in Anton Larsen Bay and Cook Bay. We used collections that were from 11 years, including 6 years before marine heat waves and 5 years that have happened since marine heat waves occurred. And from each of these individuals, we got their lengths, weights, and their otoliths. So otoliths, which these are whole Pacific Cod otoliths on the left-hand side here. These are inner ear calcium carbonate structures that grow in proportion with the fish. And they grow in these deposited undisturbed layers. And so that picture on the right is a cross-section of one of these otoliths. And you can see those rings that grow out from the core to the edge. And we can count those rings to get an idea of their daily age. And so for each of these otoliths, we counted the otoliths at least twice, with 1 to 3 readers doing that. And we only included otoliths that we could read consistently with less than 10% difference between readers. And then also, since these otoliths grow in proportion with fish, we can use information on those increment widths to get daily ages. So since otoliths grow proportionally to fish, we can create this relationship between the otolith radius and fish length. So that means if we know the length of a fish when we captured them, and then we can use their radius to determine the size of a fish at a earlier age, we can back calculate that size of that fish, and we can determine the growth rates of those fish at that time. So then we also used temperature data. We interpolated these temperatures from the GAC1 station at the surface and the bottom. So if you remember from Pacific Cod life history, individuals will hatch out demersally, and they'll spend about a few days in those demersal depths before rising to the surface and feeding pelagically. So we assumed that individuals experience bottom temperatures for about the first 5 days after hatching, and then experience surface temperatures after that. So these figures here show time on the x-axis, and then on the y-axis is the different temperature values, with each of those lines representing a different year that we used in our analysis. Now, one of the reasons I wanted to show you these temperature data is to just demonstrate that large range of temperatures that were experienced both within and between marine heat waves. So if we focus in on a couple of years, just 2012 and 2013, that we considered before marine heat waves, and 2015 and 2019, which were during marine heat waves, we can see that even within our categories, there's a pretty broad range of temperatures that were experienced. And so that means that there could be a lot of value in examining these thermal responses within these categories of marine heat waves, rather than just only looking between categories, but sort of also examining the thermal response within and between. All right, so to start out with, I'll talk about the hatch phenology piece. All right, so our question with this, we started out wanting to know whether hatch dates actually differed before and since marine heat waves. And so we got our hatch date data from those otolith ages, and we aged a large subset of individuals with each year, and then extrapolated or created a relationship between an age and length relationship given those otolith ages to the rest of the population. So this figure here has year on the x-axis and hatch date on the y-axis, with the data with the gray box plots being before marine heat waves and the light blue box plots being since marine heat waves, with each of those dots representing the actual hatch dates of individuals. And we addressed our question with a linear mixed model. And so we're interested in sort of these distribution of hatch dates, whether or not they are actually different. And so if each of those lines represents a mean, we're trying to kind of understand how those distributions differ. However, we also have data within each year. So that means that within each year, there's also its own distribution of data, and responses might be more similar within years than others. So we can actually, with this linear mixed model, we can account for the fact that those individuals within a year might be more related by using random intercepts. So our mixed model had a response variable of hatch date and the predictor variables of marine heat wave status, so whether individuals were born before marine heat waves or since marine heat waves, and then our random intercept of year. Our analysis showed that individuals did hatch earlier since marine heat waves, with the mean estimated hatch date since marine heat waves being March 21st, and whereas before marine heat waves, it was around April 9th. And when we looked at pairwise differences between years, it was an even bigger range, with almost a month and a half of a range of mean hatch dates. So the mean hatch date in 2018 was March 5th, and then the mean hatch date, I think this was in 2013, was April 17th. So a pretty broad span of time that is shifting between these years and then between the marine heat waves. So then we wanted to understand how much of this variation that we were seeing, this shift in hatch dates, was due to warmer incubation temperatures. So when embryos are fertilized, and then hatch out, that duration of time in between that is the incubation duration. And since that incubation duration is affected by temperature, previous studies have actually found a relationship where you need 110 cumulative degree days before Pacific Cod will hatch out. So then when warmer conditions happen, that incubation duration will actually be shorter because you're reaching those cumulative degrees days at a faster amount of time. And so when we looked at this, we found that warmer incubation temperature actually accounted for about 5 to 6 days in that difference between hatch dates. However, that means that there was still 13 to 14 days, or about 70% of that difference in hatch dates that was still unaccounted for. So one potential reason for this could be selection, where only individuals who hatched out earlier ended up surviving, causing that shift in distribution. Now, this could have occurred if there was a mismatch with prey. So if when the marine heat waves happened, preferred prey for the larvae actually also shifted their distribution in time as well. It could be that only the earliest individuals that hatched were able to get this prey, and thus were the only ones that survived. Additionally, earlier hatch dates, the remainder of that earlier hatch date timing could be due to earlier spawning by the adults. Numerous studies have shown that adults will actually spawn or reproduce earlier when they're exposed to warmer temperatures. So that might also explain some of this. And then finally, it could be that during the marine heat waves or since the marine heat waves, a different parental source has been contributing more to the population. And so with those individuals, they maybe reproduce earlier, and that causes a shift in hatching phenology as well. Now, this is important, the fact that they're shifting, because it could have larger implications for the populations. It could be that earlier hatchers compete better than later hatchers. As I mentioned, if those earlier hatching individuals are larger when they reach the nursery areas, they might be able to compete better and actually cause a skew in the distribution of hatchers even further. However, keep in mind that since the marine heat waves, there was or during marine heat wave years, there was a decrease in the total recruitment to the nurseries. So it could be that those earlier hatchers actually experienced higher mortality because they experienced different early life environments that caused greater mortality rates in marine heat wave years. All right, so next up, we'll be talking about size at age. All right, so our first question with size at age was actually just, did size at age differ before and since marine heat waves? We again used information from otoliths to get the ages of individuals that we caught, and we used the lengths of fish that were captured. So this figure here shows years on the x-axis, and then the standard length at capture on the y-axis, both for all years before marine heat waves and since marine heat waves. And we again used a linear mixed model. Now this time we're trying to tease apart whether the slope of that relationship between age and standard length differed since marine heat waves from before. But we also, again, have individuals that are coming from different years. So we wanted to account for the fact that that relationship might differ slightly depending on which year individuals were caught. So our linear mixed model for this included the response variable for standard length, the predictor variables of marine heat wave status, so whether individuals were born before or since marine heat waves, and an interaction with age there, and then our random intercept of year. So for this, we found that there was actually more rapid increase in size at age since marine heat waves. So this figure, age is on the x-axis, and our standard length at capture is on the y-axis. And then the blue points and line represent since marine heat waves, and the black represent before marine heat waves. So we had this interaction in this slope, meaning that individuals that were caught since marine heat waves when they were at younger ages were actually 5 millimeters smaller than before the marine heat wave. And then at older ages, they were 11 millimeters larger. So this could occur due to some plasticity, and which could have occurred with carryover effects or something like compensatory growth, where maybe those individuals that were smaller at age since marine heat waves had a higher growth rate given similar resources, which could cause them to have compensatory growth and end up being larger at age later. However, these data, remember, were just size at age. And so we are about to dig into growth later in a second. Another reason could be genetics, in that these individuals could be coming from a different source population again, where individuals that were exposed to those warmer conditions since marine heat waves might have a different size at age relationship and potentially different growth rates. Now, this is important for a population because, again, we think of the larger fish and higher condition in the summer as potentially competing and surviving better over winter, which could have larger implications for that cohort and that population overall. But again, don't forget that in the nurseries, we had fewer fish in general since marine heat waves. All right, so now looking at our early growth rates. All right, so our question with this was looking at how temperature affects growth before and since marine heat waves. And so since Pacific Cod are ectotherms, their growth rate tends to be affected by temperature. And so one way that the marine heat waves could have affected Pacific cod is that if those individuals before marine heat waves were exposed to those cooler temperatures, they might be at a lower point on this relationship between growth and temperature. And then since marine heat waves, maybe they were exposed to warmer temperatures and so had a faster growth rate because they were at a higher point in this curve. However, it could also be that the relationship between temperature and growth differed between the marine heat waves, between the periods of before and since marine heat waves. Additionally, given what we had seen with size at age, we can now sort of look at that these a little bit to see if those differences in size at age might be due to those growth responses to temperature. So we chose to address this question of how temperature affects growth with a generalized additive mixed model. So we used that because we did not have to have an assumption about the shape of that relationship with a generalized additive mixed model. So for example, if we were looking at the effect of temperature on growth, we could break the data into different sections with knots. So these dashed lines represent knots. And within that, we could have a different relationship with the data for each of that, which allows us to have a much more flexible approach for the assumed relationship between the data. We also wanted to include an interaction with age, since the way in which temperature affects growth rate could differ depending on the age of the individual. And then we also wanted to include the covariate of hatch dates. So depending on when individuals hatch, they might experience different growth rates at that time. And so that you have to think of kind of seasonality. So if individuals hatch out earlier, they might have different growths than if they hatch later because of the different environmental conditions they might be exposed to. And finally, the mixed part of the generalized additive mixed model allows us to think about different random slopes and intercepts. So since each of our growth data were from, we were looking at daily growth rates, we were taking multiple growth estimates from an individual, so we could account for that and count for the fact that individuals might grow differently at different ages, and then account for the fact that we had multiple individuals from within each year, and that individuals might grow more similarly within each year than between years. So our final model had a response variable of growth and the predictor variables of marine heat wave status and a tensor product smooth of the relationship between age and temperature. And we looked at that with differing between our two marine heat wave periods. And then we also had a smooth relationship with hatch date that also was able to differ depending on marine heat waves. And then we had our random intercept of fish ID and age as slope and a random intercept of year. All right, so we ended up finding that our growth at age responded differently to temperatures before and since marine heat waves. So I'm going to be talking about these results with color contour plots, where age will be on the x-axis and temperatures on the y-axis. And so, for example, with this example, this would mean that a fish that it's at 50 days old experienced temperatures around 8.5 degrees Celsius and experienced moderately fast growth rates. So this color contour plot is of the relationship between temperature and age and growth before marine heat waves, and the lighter colors indicate faster growth rates and the darker colors indicate slower growth rates. So if we break this into sort of sections, we see that early growth rates, so like at day 10, individuals did not actually have, temperature didn't affect individual growth rate very much before the marine heat waves. And then as they get older, growth rates get a little bit faster. Maybe there's a little bit of an effect of temperature on growth rate here, increasing over time, but still not a strong effect of temperature. And then at older ages, again, faster growth rates at that age, but there still does not seem to be too much of an effect of temperature. Now, if we compare this color contour plot to our to the growth responses since marine heat waves, we see a little bit of a different story. So looking at this early life period, we see that there's a little bit more of an effect of temperature on that growth rate, where growth is fastest at those intermediate temperature ranges. And then at slightly older ages, we see that growth rates are actually increasing at age a little bit faster since marine heat waves. And you can see that with just how close those color contour sections are to each other. And then at the oldest ages, we see that there's a greater response to temperature since marine heat waves, with, again, the highest growth rates being sort of at those intermediate temperature values. But we also just see a lot faster growth rates in general since marine heat waves. And so we can sort of pair these two responses between the different periods. And we see that there's a little bit of a change in the way that individuals are responding to temperature, or the way that growth is responding to temperature between these two periods, with growth rates being actually lower at temperature since marine heat waves in that earliest period of life, and then increasing to being faster growth rates at temperature at older ages. And then that shape of the relationship also seems to be changing a little bit, which is most sort of prominent in this figure on day 75, when there's a lower growth rates at lower temperatures that sort of increases and plateaus since marine heat waves, but almost a very little response to temperature before marine heat waves. I wanted to mention quickly about hatch dates. Don't forget about those. We did look at the relationship to hatch dates and growth in this model as well. And we found that hatch dates were related to growth rates before the marine heat waves, but not really since, where growth rates since marine heat waves were just pretty similar, regardless of when individuals hatched. But before marine heat waves, later hatchers had quite a bit faster growth rates. So what could be sort of some of the causes for these changes in this relationship with growth? One could be prey limitation, so that slightly slower growth rate since marine heat waves during that early period of life might be related to a change in prey community. So during marine heat waves, there was also observed a change in the prey community that was to smaller individual, lower quality prey, so which could have affected larval growth rates at that time. And then for this change in the response to thermal conditions, that change in that sort of functional relationship between temperature and growth, as well as the change in the way hatch date seems to affect growth rates, that could be due to some effects of plasticity with either carryover effects or epigenetics. So individuals, the way that they respond to their environment is affected by previous environments that they experience. So what happens in the larval period could affect individuals later as juveniles and how they respond to their environments. And so if individuals are exposed generally to warmer conditions, it could be that individuals that are exposed to those warmer conditions as larvae, for example, could respond differently with growth rates throughout their life and as juveniles as well. This can also occur with maternal inputs, where if or parental inputs, where if parents are exposed to warmer conditions, they might provision their eggs differently, or it could affect the eggs epigenetically and change how larvae and juveniles then respond to temperatures later. And then again, similarly to the other factors that we've seen, it could be that there's a different source population contributing to the population that we're catching. So different genetics could cause a different relationship with temperature. And then finally, it could be due to selection, where certain traits were selected for earlier on. And so when we caught them in the nursery, we were catching only the individuals that expressed those certain relationship with growth and temperature or with hatch dates. This has some important implications, because this might mean that there's a potential permanent change in the relationship between temperature and growth, which needs to be considered when we think about models or predictions that are based off of temperature relationships. If the way that individuals are growing at temperature is changing, then that might affect our predictions for how individuals and populations might be responding to warming. All right, so to wrap this all up, what was that effect of temperature? So we found that individuals were both older and faster growing. And we found that this relationship was partially explained by temperature. So we found that temperature helped explain hatch dates directly through the effects of warmer incubation temperatures. And also, we saw that the relationship between temperature and growth changed since marine heat waves, which helped explain some of the growth response. However, temperature alone can't explain all the changes that we're seeing with Pacific Cod. And we can see that by looking at our size at age relationship. So since marine heat waves, at the oldest ages, individuals were 11 millimeters larger. However, if we look at the average growth rates for the older ages since marine heat waves, and sort of add that up to figure out growth rates, that only accounts for about 9 millimeters of that size difference. So there's still some of that size difference that temperature isn't explaining. Additionally, there are some key observations that we had that wouldn't be predicted based only on the effects of temperature. So, for example, with size at age, we typically assume that individuals that are exposed to warmer conditions are going to grow faster at all ages. However, which it would be that individuals that are younger ages would also be growing faster at older ages. But instead, we saw that there was early growth was slower at temperature with Pacific Cod, and that their size at age was 5 millimeters smaller at those younger ages. So overall, we see that there's quite a few factors that are affecting phenology size and growth responses to marine heat waves. Our focus on this analysis was really looking at that the effect of temperature on individual responses. However, we ended up seeing that it's very likely that potentially other environmental conditions, things like prey and the physical environment, are also affecting that individual response, like growth and phenology. However, temperature is also potentially indirecting our individual responses by affecting those other environmental conditions, by affecting prey distributions and timing, which could cause some interactive responses between the effect of temperature and environmental conditions on those individual responses. And then additionally, previous experiences can affect some of those more immediate responses or those immediate experiences. So things like epigenetics and carryover effects, those are likely affecting the way that individuals are responding to their current environment, both temperature and those other environmental conditions. And then things like the population composition is also probably playing a role with selection and parental composition. So this is a lot to kind of tease apart. And so we have this need to try to understand some of these more complicated inter and intra-individual responses to the marine heat waves beyond just the effect of temperature. So that is the next steps with this project, is to really look at those other environmental conditions and try to see how those might be affecting our growth responses in early life. So with that, I'd like to thank everyone who helped out with this project, everyone in the Miller Lab, and particularly Thomas Murphy, who did a lot to prepare otoliths that we were able to use. I'd also like to thank NOAA Alaska Fisheries Science Center, as well as everyone who helped collect samples across the years, because without that long-term data collection, we certainly could not have really looked at these questions. And also, I'd like to thank our funding source, North Pacific Research Board. And with that, I will take any questions.
All right, so we covered a lot of data very concisely. Thank you very much, but we'll open it up to questions. I'm going to start with any questions online. Okay, for folks online, go ahead and put your questions in the chat and we'll work through them. And then we've got a question in the room. You'll probably have to slide the mic on.
Is that on? Yeah. Thanks, Zoe. That was great. As you know, I just read the manuscript for this, so it struck me at the time, and it struck me again in your presentation. One of the things that struck me a lot was the fact that in the pre-heat wave era, there actually was the lack of temperature response in growth rates of the larvae. And I guess my question is, did you expect that to be the case? And if you did or didn't, like, what was do you have any ideas for why that might have occurred?
Yeah, no, I did not expect that. I definitely expected more of the responses that we assumed we'd see, where, that there's that nice little growth curve where increasing temperatures cause higher growth, at least until some point. Yeah, and so that was a bit surprising. I don't entirely know what is going on with that. And it, it could be like, so we took temperatures from like a pretty broad area and sort of assumed individuals were experiencing those, which is like relatively defensible, but it could be that individuals are actually experiencing some more microhabitats and different things there. So that could be one cause for it, but it could also be that just temperature isn't the limiting factor with growth in that case. So it could be that other factors were affecting growth rates more than temperature, and whether that might be like prey environments or something else, that would be my guess as to what's going on there. But yeah, I would love to hear if you had any thoughts about it too. So.
So one of the causes, at least of the blob, wasn't heating, it was actually lack of cooling, right? So the ocean did not mix.
Right.
And therefore the surface waters were very warm, and they're also very, very low nutrient. And I was just wondering if you've thought about that, and that it's temperature and prey and lack of nutrients. So this normal production and really whether, it seems like from a lot of your talk, you're assuming that these guys are well fed. The growth is really regulated by temperature, not prey availability. And I was just wondering if you've thought about that and what your conclusions are.
Yeah. I mean, that's a great point. Like you can't have growth if you've got great temperature but no food. So that's sort of like, we sort of were like, the first piece we want to focus on temperature, just because, we've got this data, we can really dig into that temperature question. We have some expected relationships with temperature. Let's assume food isn't the limiting factor, but it is, like we know that the marine heat waves affected the prey dynamics. So our next steps are to really look into that and look at some of the prey communities and dig into some of the, great like zooplankton databases that are out there during those years and before those years, to sort of see, can we explain some of the remaining growth variation with the prey dynamics, and maybe potentially try to tease apart if there was some interaction between temperature and prey as well. But I mean, that kind of speaks to Tom's point too, is it's very much that maybe we didn't see in the before time that response to temperature, because prey was the limiting factor. It wasn't really temperature. And then once we have temperature and prey limiting, that's when we're in sense the marine heat waves, that's when we're starting to see some of that, more of that temperature response as well. So I do think prey is probably explaining a lot of that remaining variation, but yeah.
Any other questions? Looking online real quick.
Okay, we have one question. SS temps rose during the marine heat wave. Were the bottom demersal temps also higher, or were the cod hatched in cold water and then migrated as juveniles above the thermocline to marine heat wave influenced warmer water?
Okay, I think I got all that, but I'm gonna take a look at that just to make sure since I
Okay.
have the ability to look at that here. Okay, so surface temperatures rose during marine heat waves. So yeah, what we did see that when there was a marine heat wave in the surface, there was also a marine heat wave in the bottom water temperatures. So those were also warmer than normal. So it wasn't a situation where individuals hatched into a similar environment that they had before the marine heat waves and then kind of warmed into those warmer waters. Definitely surface waters were warmer than the bottom waters, but the bottom waters were warmer than they typically were during heat waves as well. Does that make sense? Please let me know.
Yeah, feel free to put a follow-up in the chat for that person who asked that question, but we're going to get a question in the room while we work on that.
Thanks, Zoe. That was awesome to see all put together. I was curious, so in the graph where you're showing the hatch phenology, and I'm not going to make you go all the way back, but to me, it looked like you actually had just a decreasing trend or so earlier hatch with time.
Are you talking about this?
The one before it.
Oh, okay. This one?
Yeah. So
Yeah.
like if you had everything the same color,
Yeah.
what I really see is just a change in phenology over time that doesn't
Yeah.
seem to be marked by a marine heat wave,
Right.
but that because the marine heat wave is happening later, it is showing up as
Right.
arriving earlier hatch. So I was curious if you kind of looked at maybe like other drivers of phenology or other metrics to kind of validate that on like a different null of something
Yeah.
else going on. So
I mean, I think that's a great observation. And that was something that kind of struck me too when I was looking at the data before analysis. And so what I don't know is if like, if we just, oh, I'm using a pointer on the screen, I can see. So if we just look at the before marine heat wave years, it's hard to tell if there was a trend of like earlier and earlier hatch dates or not. So that's what I don't know. I can't tell you that. But I do think that sort of like since the marine heat waves, it does look like it's getting earlier. And that's brings into the interesting question of 2017 and 2018. So 2017 and 2018 had like cooled down again. However, a lot of the trends that we were seeing were still some of these remnant trends since the marine heat waves, with some of our earliest hatchers being in years that actually weren't considered marine heat waves. And so it could be that hatch dates are getting earlier and earlier. It could have been triggered by the something going on in the marine heat wave, but it could be like, this is where I think we have gives me the stronger inclination that something like a different source population is potentially contributing to it, and or a difference in the spawning population in some way, somehow. So like a study by Lauren Rogers found that I think it's older individuals tend to reproduce earlier. And so it could be that during those years of the marine heat waves that a lot of the younger individuals died and only older individuals were contributing to the population. And so it could be that because of that, there's sort of this like older individual bias that we're continuing to see because those younger individuals didn't make it to reproductive age. And so hypothetically, if things return back to normal, we should see that like maybe return back, or there's this, shift in the population in general. So yeah, it's very interesting, and who knows what's going to happen next. And I'm very excited. Like I want more years of data. So
So we do have genetics on the source populations.
we not for every, we don't have like for every year, but we do have for more recent years, if I'm correct. I don't remember, when did they start collecting? Sorry, I'm looking at Jessica.
Yes, there's some samples being analyzed for 20 and 21, and 19 might be included in there, and a greater spatial distribution, but we don't have any of those results yet either. And I guess, well, I'll just give you a cue. What about what are we seeing in 2021 that gives a hint to Emily's question?
Yes,
Worth
yes.
mentioning.
So in 2021, the juveniles that were caught in the nursery had this really interesting size distribution where there was a lot of individuals that were very small and a lot of individuals that were very large. And so there's this like 2-peak size distribution. And so that makes us think that there could have been like 2 different spawning events or 2 different populations maybe contributing that to the population as a whole. Whereas like, and so then maybe before the marine heat wave, only the individuals that were spawned later or reproduced later ended up making it. And then something happened in the marine heat wave that we're seeing both of them now. So lots of different questions about that. But yeah, so we're hoping with 2021, especially with some of those genetic results, to see like, are these 2 different populations that are ending up here? And we've just previously seen one of them where, you know, so yeah.
Stay tuned.
Yes, yeah.
I think we have a follow-up online. Go ahead, Rose.
I have another question. Just curious if fishing data has been considered.
So sort of in like the sizes of individuals caught or the number caught, or what kind of fisheries data?
Okay,
yeah,
we'll give people a chance to kind of respond in
Yeah.
chat. And it seems like we had another question in the room.
I had a question about temperature, and you've mentioned a couple times different populations, and the assumption I was assuming is that these different populations are experiencing different temperature than what your GAC1 temperature. And I was wondering if you have any knowledge of really fine scale temperature variation within that area. Was it everybody was hot, you know, when you had these heat waves, or was there, especially around Kodiak with all
Right.
those little bays and everything,
right.
do you have any sense of how much variability and whether your GAC1 temperature is really capturing
Yeah.
what was going on?
Yeah, so I know, again, Lauren Rogers has done a lot of great work. She did an analysis that sort of compared GAC1 to other regional estimates. And as far as like regional estimates, GAC1, even though it's like a 1 point, actually did a pretty good job of capturing regional estimates. But what I don't know is how much like sort of variation within the region could be contributing to that. So I feel like someone might know that. I am not that person. But yeah, that's like a really good point. You know, how much is, you know, spawning population here, even, you know, relatively close, like how much variation are they experiencing? So I'm not entirely sure. Ben and Lauren have also looked at that spawning habitat distribution, and given what Ben has seen in the lab, the hatch success is really narrowly related to temperature. So it's a very narrow range where you get a decent amount of survival of eggs during incubation. And so it could be that there's a big variation between what spawners are experiencing, but supposedly only the ones that have experienced this very narrow range of incubation temperatures are actually going to survive to hatching. So it could be that they're coming from different areas, but they can't quite experience, like what we're seeing at the nursery can't come from too much of a big difference in temperatures, just because of what we think is happening for survival from egg to larvae to juveniles. But yeah.
Okay, follow up online.
I do have a follow-up for the fishing data, commercial and long line and trawl.
So with using some of those data have been considered in things like the stock assessments and the overall population predictions, but I don't know if that has been necessarily used to look at the early life component yet. I think it would be interesting to sort of use that to try to do some of like, is the population moving a ton? But I also know, like I've heard that since the fishery closed in 2020, it's back open, but a lot of people aren't returning to the fishery in Kodiak, at least, is from what I've heard. I thought. But yeah, I guess I'm not entirely sure what you mean by how they've used that data. Those data are used a lot in the stock assessment, and the stock assessment is trying to incorporate some of the early life history components as well too, as what's going on in like the nursery grounds. Yeah, I probably did not answer your question. I'm sorry, but
So Zoe, can you go to your last slide where you had your contact information so that if you do have a follow-up question, you can reach out to her directly, and that might be a more efficient way to get your questions answered for those kind of details. Any other questions in the room? Rose, any other questions online? All right, I think we get to let Zoe off the hook, but one more round of applause for Zoe, please. All right, everybody. Thank you for coming. I hope to see you next week. And Zoe, again, thank you. And thank you. Good luck on your next steps. We'll miss you around here. Okay, okay, for folks online, we're going to go ahead and end the presentation. Thank you so much.